Good afternoon. I think I'm the last presentation of the afternoon before lunch, so I'll keep this moving along. So Immpact Bio is the first and only dual-targeting CD19, CD20 CAR T-cell company for the treatment of both hematologic malignancies and cancers, as well as autoimmune diseases. We focus really on multi-targeting. Our lead programs are all dual-targeting programs. We do in-house manufacturing. It's a differentiated manufacturing platform in the sense that it makes cells that are actually heavy central memory type cells, which have a different profile than T effector cells. And we have several clinical programs right now, both in the heme malignancy side as well as in the autoimmune side, and a first-in-human study planned for 2025 for solid tumors. So our pipeline at a quick glance, majority of this presentation will focus on our bispecific CD19, CD20 approach for heme malignancies as well as autoimmune conditions. One program is actively recruiting right now, and multiple patients have been dosed. Data looks very favorable. And another program is about to start enrolling. Sites have been activated in lupus nephritis and non-renal SLE. We have 2 INDs opened, 3 fast-track designations, and about 8 weeks ago, the California Institute for Regenerative Medicine gave us a competitive clinical grant for $8 million to conduct our lupus nephritis study. We have additional protocols opening up ex-US, 3 indications, a basket trial being done in multiple centers, and this will be used to generate data that will then translate to other markets. In addition, in 2025, first-in-human trial in solid tumors, starting with gastric and GE junction cancer. A dual CAR in our sense is actually a tandem CAR. It's a single polypeptide. In our lead program, it's a CD20 binder in the membrane distal position and a CD19 binder in the membrane proximal position with a 4-1BB co-stimulatory domain. As you know, 4-1BB has just a favorable profile when it comes to CAR T-cell persistence, as well as in terms of cytokine release. In addition, CD20 is a very resilient target, as you know from the data with multiple lines of rituximab use. CD20 as a target antigen just pretty much stays at the surface for most patients. So this dual targeting reduces the chance for antigen escape. In addition, I mentioned our proprietary manufacturing platform. When the leukopak comes in, we do magnetic bead isolation for CD62 ligand cells. CD62 ligand is a marker on more stem-like or naive T cells. And then by the time we finish our manufacturing process, our cells are very heavy central memory cells, which tend to also have a more favorable safety profile, but they are noted with better engraftment, improved persistence, and as well as reduced potential for exhaustion. UCLA conducted a trial started in the second half of 2019. This was the technology we ended up in licensing to Immpact Bio, and that particular technology was taken into patients with a mixed histology, non-Hodgkin's lymphoma, all clinically aggressive disease, and they had to have failed at least 2 lines of therapy. The study continues to enroll at UCLA. Immpact Bio has opened its own study under our own IND in aggressive B-cell lymphoma, but at a snapshot, this is what the UCLA data is as of July 2023, presented at ASH 2023, and it has years of follow-up at this point, a 73% durable CR rate, median PFS has not been reached, despite median overall survival follow-up has been greater than 32 months as of last year, so it's actually longer now. And no neurotox has been observed in this program, and grade 1 CRS or no CRS. So very favorable safety profile. From our vision, this can become an outpatient therapy, and the early signs that we're actually seeing at Immpact Bio point in that direction. So I'll go into aggressive B-cell lymphoma program and then to the SLE program. So CD20, just not to belabor the point, is a very resilient antigen. CD19 and CD22 are very good antigens also, but they do get lost with selection pressure when you actually target those particular antigens. So this combination of 1920 gives a potential for reduced antigen escape. This is just the UCLA data with overall survival and progression-free survival curves. Median PFS has not been reached, median overall survival has not been reached, and I've already mentioned the current safety profile. In Impact Bio, we started the study in this third quarter of last year. We have, as I mentioned, dosed multiple patients at this point. We're looking at both CAR-T experienced and CAR-T naive. Second line, they have to fail at least 2 lines of therapy, so third line plus patients. We've had very good success in manufacturing with a 17 to 21 day turnaround time. We do our own manufacturing in Los Angeles in our own facility, and we do all our release assays in-house, so that helps with the turnaround time to the centers. We use a standard conditioning regimen that's noted in the bottom of the slide, not a high dose one. And we do lower the dose for conditioning when we actually dose CAR-T experienced patients, as well as lowered conditioning regimen when we dose autoimmune patients. There is an inpatient period, but now we have openness to in the centers for outpatient therapy. The FDA IND amendment has passed for that. We're enrolling in multiple sites. This is an example, a clinical vignette of a patient, 35-year-old female who had Hodgkin's disease as a teenager. She actually then developed non-Hodgkin's disease in her 30s, very aggressive non-Hodgkin's B-cell lymphoma, DLBCL, got 2 lines, up to 3 lines of therapy, ended up getting 100 million CAR-positive cells with standard conditioning regimen, 21 days door-to-door time, and both the target lesions and non-target lesions all disappeared in 28 days. No CRS noted, no ICANS noted. This is an example of the perfect patient, and this is what you want to see over and over again, and she continues to stay in CR on longer follow-up. The study that's ongoing is actually a multi-cohort study with greater than third-line CAR-T naive, the CAR-T experienced population, as well as a second-line CAR-T naive population. We will make a decision by the end of this year, early next year, which cohorts we want to take forward. A lot of it has to do with feasibility, trial speed, and other aspects that we want to, which cohorts we want to take forward. And then we have at least early thoughts and feedback around what would be acceptable from a registration standpoint. The market would obviously be the most opportunity would be in second line, and that's what we're ultimately aiming for. So now moving on to IMPT-514. The previous program was called IMPT-314, which was for aggressive B-cell lymphoma. IMPT-514 is for the autoimmune conditions. While our first indication is lupus, we're actually working on additional INDs at this point. So everyone knows the data that came out of Erlanger Institute in Germany, so I'm not going to belabor that, but truly transformative data, again, shown in the New England Journal of Medicine this year. Patients continue to do very well. The data is quite pristine, so I'm not 100% sure whether everyone else's data will be as pristine, but I think that it shows that there is a huge potential for unmet need in the autoimmune market, and I think there's more than enough room for more than one player in this market, frankly. Patients definitely need a therapy like this. So we're focused on the high unmet need areas targeting B cells, but I'll show you about targeting T cells also in a moment. We're looking to elicit durable responses. We have high durability in the non-Hodgkin's lymphoma with very potent B cell aplasia. And we have very good coverage for extensive autoreactive immune cells. And the proprietary manufacturing has clearly shown just a safer profile in our product. We are now doing an ex-US partnership, I said a basket trial for more cost-effective and faster data generation across multiple autoimmune indications. And in the US and other developed markets, we're actually doing a lupus trial in both lupus nephritis and non-renal SLE. This tells you a little bit about the biology of why we want to target CD19 and CD20, because people do ask us this question, well, CD20 is nested within CD19. Why would you want to target both? I mentioned one of the rationales. CD20 is a very resilient target, and in our hands, what we have seen in our labs with patient leukopaks, we're seeing CD20 is actually 5 to 10 times higher in antigen density than CD19. We've seen B cells that have very variable CD19 levels, and in both the heme malignancy side as well as the autoimmune side. And so we are targeting CD20 along with CD19. So the CD20 is also very highly expressed in what are called inflammatory age-associated B cells. These B cells are tissue resident. They're hard to get to, frankly. I mean, if you do not get a very deep B cell response with this CAR T-cell therapies, the likelihood of it coming back is pretty high. And you've seen that with years of rituximab treatment and other anti-CD20 treatment that people do relapse despite actually treating these patients. So we hope that we get this deep response because we end up getting all the tissue-resident cells also. And importantly, we spare the plasma cells. I think plasma cells do have the potential to cause disease, but the long-lived plasma cells are there for a reason. They give you the coverage of immune coverage for vaccines that you've gotten previously, as well as for previous infections. And the Schett data also showed that patients retained vaccinal memory even though they got a CD19 CAR T-cell therapy. In addition, there's a T-cell compartment. This is emerging data over the last several years. Both CD4 and CD8 positive T-cells do have a dim level of CD20, and we've demonstrated that in-house also with patient leukopaks coming from a variety of autoimmune diseases, and we have seen these CD20 positive T-cells. And these T-cells are considered dysfunctional T-cells associated with high levels of pro-inflammatory cytokines and actually play a role in disease pathogenesis. So that's an area that we are studying more intensely to see if our CD20 targeting also reduces the CD20 dim T-cells. Across the multiple indications that we have done in-house, both process development as well as research work on, we have shown very high transduction efficiency, very good fold expansion, despite the fact that the donors are actually on active immunosuppressants. We see B-cell targeting as well as IgG secreting B-cell targeting. And importantly, on the right-hand side, you'll see the blue curve in the middle. That blue curve shows you that because of our manufacturing process, and we've seen this, by the way, across non-Hodgkin's lymphoma as well as in the various autoimmune indications, our cells just generally produce less amount of gamma interferon, just a lot less compared to the traditional manufacturing, which we believe is one of the reasons we're seeing the safety profile. This is our registration pathways we're thinking about right now. We're starting with refractory active lupus nephritis, and then with the safety per dose level, we can open it to refractory severe SLE across non-renal SLE also. And then the 3 additional indications that are being done ex-US are going to start enrolling by the middle of this year. And we have fast track designations across this and a few more INDs coming along in this area. Manufacturing at a high level, very straightforward. Just a simple linear process, very robust, very consistent. We produce batch after batch this way. And it's an 8-day process at this point. We can extend it longer to make dose. In the autoimmune side, it does not have to be extended any longer than 8 days. We can easily make dose. On the oncology side, the hemalignancy side, we do have to extend it for some patients, and other patients we can make it within 8 days. But we have our own state-of-the-art facility. We are producing mostly central memory cells with some naive T cells in there, very similar to the UCLA product. And again, very rapid turnaround time for the sites, which they really like, because they don't, in the oncology side especially, they don't want to put patients into bridging therapy if possible. And then we're switching over to an automated manufacturing platform in the second half of this year. So before we enter into any pivotal trials, so we don't have to make any CMC level changes in the pivotal state. Very quickly on our TGF-beta platform, we're really looking to convert the immunosuppressive tumor microenvironment for the solid tumor side. TGF-beta is a very potent chemokine that just inhibits other immune cells, including T cells. We have what is called a switch receptor. It takes an inhibitory chemokine, TGF-beta, converts it to an activating signal for the CAR T cell, and then we couple it actually with another binder for tumor-associated antigen, just to target specific cells. And we have a lead asset at this point going into gastric and GE junction cancer. It's a well-validated target. And there's a number of tumors, solid tumors, that can both have TGF-beta signature as well as a very specific tumor-associated antigen that you can actually target by this mechanism. And we have shown in our lead asset, IMPT-601, very robust anti-tumor activity. I think the most important part of this graph, other than safety, is actually the middle side. We have shown in the tumors, and because both our targets in this case have a 100% homology between murine and humans, this is pretty validated approach. We have seen infiltrating immune cells in the tumor, the solid tumor, when we treat with IMPT-601. That means the innate immune system and the immune system of the mouse is being recruited, and epitope spreading is happening within that particular tumor, which I think is going to be important in solid tumors just because of the diversity of cells and the various mutations that occur in solid tumors. So we're doing IND-enabling studies now, and then we'll be initiating studies in the clinic in 2025. Multiple inflection points in our portfolio. This is a truly multiple shots on goal strategy. I'm only focused here on the CD19, CD20 platform. No mention here of the solid tumor platform because we got to take that into clinic first. But multiple data points are coming up, and you can see here that we have several FDA interactions also coming up as a result. I will say health authorities are very excited about this field and the potential to continue moving the bar up for the treatment of patients with these high unmet needs in both heme malignancies as well as in various autoimmune conditions. Thank you very much for your time. We're very excited. We're a young company. We didn't exist 2 years and 3 months ago. I joined the company. It was a small research lab, and today we have over 100 people, dedicated employees making incredible products for patients and treating patients with patients we're seeing getting complete remissions. Very, very happy to do that for patients. Thank you very much.